Ladies and gentlemen, good morning. I'm Jim Byron, President and CEO of the Richard Nixon Foundation. Welcome back to the Nixon Library as we start day 2 of the Nixon National Cancer Conference. I want to thank our board members that are here in the room with us, and I want to also thank the NCI Cancer Center directors. There are at least 5 running around here somewhere who have flown in from all over the country, so thank you very much for being here. Last evening, we were treated to a rousing discussion between industry leaders about the mathematical underpinnings of cancer and how and why we understand cancer today as we do, and how that can help scientists to develop a cure in due time. And last evening, in a video from 1972, we heard President Nixon say that you cannot separate one disease out without having experts in other fields making contributions. So we're going to cover a host of topics today in keeping with that theme. We begin this morning with a program on regenerative medicine and stem cell therapy, perhaps one of the most interesting and rapidly evolving aspects of modern medicine. And here to lead this program is Dr. Donald Skip Trump, founding CEO of the Inova Schar Cancer Institute and Inova Health Systems, and a professor of medicine at the University of Virginia Cancer Center. He is also the co-author of the new book, Centers of the Cancer Universe, A Half-Century of Progress Against Cancer, which I will add is for sale in the Nixon Library's gift shop. Doctor?
Well, thanks very much for the kind introduction. I'll try to restrain myself from advocating that you stop in the bookshop and buy a book. I'm really delighted to be here, and most delighted to be here with a panel of distinguished colleagues. This is something I've been looking forward to, and hopefully we'll have a discussion this morning that really does outline where we started, where we've been, and what the future might hold, with a particular emphasis on cellular therapies. In keeping with the historical theme that this conference embodies, I think it's interesting to reflect on the history of cellular therapy. In 1957, Don Thomas in the New England Journal reported the first stem cell infusion in humans. 6 patients were treated after chemotherapy and total body radiation. 2 patients had a transient graft. They all died, but that established the feasibility of stem cell transfer, and we've come a good distance from that period of time. Don Thomas did that at the Mary Imogene Bassett Hospital in Cooperstown, New York, working for Dr. Farabee, who had recruited him there, both Farabee and Thomas having been at the Brigham Women's Hospital in Boston. Fast forward to 1990, Don Thomas and Joseph Murray received the Nobel Prize for their contributions to the understanding of transplantation. Dr. Thomas for his groundbreaking work in establishing the feasibility and success of stem cell therapy, Dr. Murray for his contributions in renal transplantation. Interestingly, both Murray and Thomas had a Brigham and Women's background as well, so one might argue that Brigham and Women's has a foundational position in the field of transplantation. On December 8th, 1990, December 8th is today, you'll recall, Dr. Thomas gave his Nobel Prize lecture in receipt of his Nobel Prize. And I would commend that to all of you who are interested in stem cell therapy, because he does review how he got to the 1990 event and the contributions he made. Fast forward 32 years, boy, have we made a lot of progress. And what, among the things we'd like to talk about this morning are the contributions and the future of, I guess broadly speaking, regenerative medicine, but also emphasizing issues of access and the challenges that we have yet to overcome, not to mention the challenges scientifically. I'm delighted to be joined this morning, and I'll introduce each of our panelists because they're going to do the hard work, To my immediate left is Dr. Stephen Grupp from Children's Hospital in Philadelphia, Dr. Linda Malkas, Dean at the City of Hope and a drug discovery scientist, as you'll hear on her own right, Dr. Stefan Turia from University of California, Irvine, and joining us electronically is Dr. Robert Winn, Director of the Massey Cancer Center at Virginia Commonwealth University. So thank you all for being here, and without further ado, I'll turn to Dr. Grupp and ask him to tell us what's happened since 1990 in the field of cellular therapy. Stephen, one of the pioneers in cellular therapy. Stephen?
Well, thanks for that. And, you know, I think to just continue on your theme of cell therapy, and what we're going to talk about, I think, to a certain extent, is engineered cell therapy, which is, you know, genetic modification of cells. But you start with the stem cell transplant experience. So we go from Don Thomas starting this off to almost, I think, over actually now 1 million people having received the transplant worldwide. And so that is the original cell therapy. And all of us, I run the bone marrow transplant program at CHOP, and obviously we're all big fans of transplant as a modality. But the fundamental thing that you're doing in a transplant, obviously, is you're doing a transplant and you're taking cells from another person and giving it to a second individual. There are risks associated with that. And one of the dreams of a lot of us in the field is there a way to do this with the patient's own cells and avoid the risks of cell transfer, which can, in some fraction of patients, be fatal, and in the beginning was fatal most of the time. And now it's not like that. You know, the risks of fatality are low, but they're not zero, and we'd like to avoid those risks. So fast forward to 1990, at that point, they're just beginning this notion that you can genetically engineer T cells, and we talked about T cells yesterday and NK cells and cells of the immune system that are capable of actually maybe destroying cancer if you just force them to do that. And so the beginnings of the CAR T revolution are really in the late 80s, early 90s. And what's interesting about that is that nothing really happens that looks all that great for 20 years. We had to learn how to make the cells right. We had to learn how to keep the cells happy while we're doing the manufacturing process outside the body. And those are all things that were learned in that subsequent time frame. But then all of a sudden around 2010, so 20 years later, all of a sudden, you talk about breakthroughs, this was a breakthrough. So we went from nothing works, and by the way, and no toxicity because it wasn't working, to, wow, what just happened? And certainly our experience at Penn and CHOP mirrors the experience of other places, but starting with Penn, treating the first 3 patients with the adult disease CLL, something I've never seen, seeing 2 of them go into remission that lasted for many years, and then our first experience with kids, including a patient, Emily Whitehead, who I'm allowed to say it's not a HIPAA violation because if you Google CAR T, it's kind of the first thing that comes up. And by the way, Sid Mukherjee, who wrote a great book called A Song of the Cell, which just came out, wrote a terrific article about the development of cell therapy in the New Yorker a couple of years ago. And if you Google that New Yorker cell therapy, you'll see it. And it's a terrific article. But so we got to a point where we could actually do this. We could see, using cells made in at a university, that you could see patients go into remission. That first patient that we treated had significant toxicity that lasted about a week after her cell therapy. We learned how to deal with that. It's a harrowing story for the family and a harrowing story for the group at CHOP who actually treated her, including myself. But it worked, and then what we learned from that applied to the 15,000 or so people who've gotten CAR T since then. So there's definitely a learning curve on this, but we got to the point where we were seeing very significant, like 90% type responses in ALL, and seeing in acute lymphocytic leukemia, and seeing those about half those patients, not all of them, but about half those patients staying in remission for a long time. And so that's really where the whole idea of engineered cell therapy really gets a boost. So you're taking T cells from the patient, it's a big deal. You're genetically engineering them with a virus, putting in a new gene, forcing the T cells to go after the cancer. And so that looks great, but it's all, by the way, and I think you guys know this, it's all in blood cancer, leukemia, lymphoma, myeloma, where we're seeing really great results, differing from disease to disease, but not happening in solid tumors yet. We're not there yet. So we still need that subsequent breakthrough. So that's the T cell world. And now that's cancer, and of course we're talking about the Nixon Cancer Act, but like NASA, there's stuff that comes out of that that then applies to non-cancer treatments. And so the final comment I'll make is about, back to the whole stem cell transplant issue, is we love to turn transplant into something where you're not getting cells from another person. We're not there yet. But what's odd, and this is not intuitive at all, but for pediatric transplant centers, not adult transplant centers, half the transplants we do now are for non-cancer indications to fix problems, for instance, like sickle cell disease. You can cure sickle cell disease with a transplant. There are risks associated with it. Happens, for a small minority of patients. Getting a donor is a challenge. But you can imagine that you'd like to extend that cure. And what's happening now is taking not T cells, but stem cells from the bone marrow, as many of you know, genetically engineering those using similar or quite different techniques, and then giving those cells back to the patient, their own fixed cells. And that has the potential to be curative. There are patients who are years out from these sorts of treatments who seem to have stable resolution of their blood disorder. And we're very excited about that for blood diseases like thalassemia or sickle cell disease, and there are going to be 20 more of these coming along. And so I think in the stem cell and regenerative medicine world, that's the next step forward. There's been a lot of discussion about regenerative medicine outside of engineered cells, and I think that's still way too early. We'll see where that goes. Maybe we're at the beginning of that process. But the final point is, we went from treating Emily Whitehead in 2012 to the first CAR T-cell FDA approval in 2017. So that part went pretty fast. But you have to remember the 20 years that went before that.
That's a terrific summary, Stephen, and you raise many of the questions we will expand on as we go. But Linda, Stephen summarized the process of investigator-initiated science in the cellular world. Maybe you could talk about your work in the small molecule world, because it all relates.
Thank you. So yeah, I think it was the year 2000, Time magazine had a cover with Gleevec. It was like, I don't know if they said the magic bullet. We talked about magic bullet last night, but it was like huge, and it really highlighted the transition. Dan Von Hoff is a friend and a mentor to me, and he said, Linda, when I was first starting out treating patients, and we were trying to find things that would treat patients, he said, they were sending me things like plant extracts, to look at. But wow, have we come so far now. We are able to start really interrogate the differences between malignant and non-malignant cells, and that's giving rise to a whole host of new thinking and actually new therapeutic approaches targeting the phenotype of cancer versus a non-malignant cell. So, I do a lot of NCI study section work, and I've watched the growth of, targeted therapy as they talk about it. Probably sometimes half the grants that are coming through a particular study section are really based on finding these new molecular targets and then trying to exploit them for the treatment of cancer. So my own journey, and I have to thank the NCI for this. When I was in the last century, the last millennium, when I started my research program, I had put forth an idea to the NCI into a study section, and I said, I think DNA replication is altered in cancer cells. And it didn't fly too well in the study section, but the NCI said, we are really interested in your idea, and they actually awarded me a director's award, and it launched my career. But with those resources, I was able to interrogate it. It started in breast cancer and then went full course into neuroblastoma, children's cancer, really looking at the change in the active DNA replication in malignant and non-malignant cells. And yeah, things were different in cancer cells. Cancer cells corrupt the active DNA synthesis. From there, we discovered a target, and I actually moved to City of Hope 10 years ago because of their a really unique mindset on the part of the institution to really drive translational science and their commitment to it. So in 10 years, I went from showing up with, I've got a brand new molecular target that even drug companies told me that because it didn't have an enzymatic activity, that it was actually, it had a protein-protein function, that it worked in protein-protein interactions, that it was undruggable. 10 years later, I'm very happy to say that this past September, we actually went to a phase 1 clinical trial with a brand new drug against a brand new molecular target. And my story just highlights what is going on in the cancer therapeutic space. I mean, it's incredible technologies on the cellular fronts, finding precision medicine, identifying patients now, what's unique to a patient. I mean, we used to, 20 years ago talk about, maybe someday, we'll be able to, a patient comes in, as opposed to putting them on a standard protocol. Everybody, if you got breast cancer, you go on this particular protocol. Maybe it's not going to be that way anymore, It's going to be subtype, subtype, subtype. And, the leukemia space really shows that. So I just wanted to give a general sense that there is an incredible amount of excitement around the better defining of what is malignancy versus normal or non-malignant, and that these differences are now being heavily exploited for new therapeutic approaches, small molecules, cellular, biologic. It is a new day.
Thanks, Linda. And Stefan Turia is a cellular therapy expert. The stage has been set, Stefan, of what the directions and the progress we've been made. Maybe you could comment on your work and also some of the challenges in the future.
Thank you so much. I am actually indeed a stem cell transplant and cell therapy physician. I moved from MD Anderson Cancer Center about 3 years ago to UCI. And my goal is not only to build the transplant program here, but to continue my research in NK cell therapy. And we're very excited about this field. Many believe this may be the next important cell therapy that may be approved. Our work began more than 10 years ago at MD Anderson. I was there for about 12 years. And we started to expand NK cells. We realized that everyone has NK cells in their blood. It's one of the main anti-tumor, anti-cancer cells that will fight modified cancerous cells that occur constantly in our bloodstream. And we started to expand this because we realized that the number of cells we have, NK cells we have, or we can extract from one's blood is too small. So to obtain a therapeutic effect, and the effector to target ratio, which is probably too skewed towards a tumor cell. Tumors are characterized by incredible proliferation of cells and an extraordinary expansion in one's organism. So we focused on expanding these NK cells to potentially target tumors. So we put them after transplant. And I can add a little bit to what Dr. Trump was mentioning about stem cell transplantation, which I'm very passionate about. And stem cell transplantation took off, in fact, when Dr. Donald Thomas published a paper in Blood in the 90s, I believe, not sure if it's, I don't know exactly, I don't remember the date, but it took off because he treated 100 patients with leukemia, and about 10% of them survived. Probably if we would not have seen 10% survival, I'm not sure the field would have taken off, and we have gotten from stem cell transplantation to identifying cells in the blood with therapeutic benefit. And of course, the next major breakthrough was T-cell therapy. Now, you've probably heard yesterday in the news TIL therapy from Dr. Patrick, from Moffitt, that may work in melanoma. But continuing the same line, we focused on a different cell in the blood, which is NK cells. And we were very excited to see that we were doing stem cell transplants, and we intuitively put these cells after transplant because we wanted to prevent disease coming back. And after transplant, which in their rate of relapse depends a lot on what we treat, what disease we treat. And there are diseases, for example, high-risk acute myeloid leukemia, relapse rate would be in excess of 50%. So we said we really need to make improvement. There are types of leukemia that we transplant, but the relapse rate can be even higher than that. So we've administered these NK cells after transplant. We found extremely low relapse rate. We treated 24 patients in a paper that we published, and we saw extremely low relapse rate of 4%. And actually, one patient relapsed who ended up not being eligible for the study because had malignant cells in the blood but not in the bone marrow. So it was somewhat missed that this patient had 6% blast in the blood, and we ended up being treated, ended up being counted. But in fact, the relapse rate in eligible patients would have been 0. So this was, I think, was extremely exciting because we might be able to significantly enhance the power of hematopoietic stem cell transplantation. And this made us think that maybe we can apply NK cells outside transplant in patients with very advanced leukemias that have very dismal prognosis in general. And we've looked at a group of, in a phase 1 trial of, we treated about 12 to 15 patients with a refractory acute myeloid leukemia, refractory to median 5 lines of prior therapy, and we had a 50% response rate, complete response rate in this group, and we're trying to publish these results. And we were trying to expand this. A fraction of these patients were able to get to transplant because you don't get to a transplant unless you have disease control. So taking a fraction of these patients to transplant in itself was a big deal in our minds. So I'm really excited about continuing this work at UCI. We're trying to, I put the transplant program together first, and I'm trying to get to the next step, which is NK cell therapy. Hopefully, we'll expand to other tumors where we, as compared to T cells, and Dr. Grupp really pointed out that the CAR T cells worked extremely well in these B cell malignancies. However, it seems like our ability to expand has kind of plateaued. We really need to find the next breakthrough to be able to move to a different type of cancer, different setting, and I'm hoping that with NK cells, which have natural anti-tumor properties, we could potentially find a niche, at least, for this group of, for NK cells, and which will give us the opportunity to make a difference, at least in one area of cancer therapy.
Well, that's terrific. Thanks, Stefan. And you've heard from my colleagues the scientific challenges and hurdles that have been overcome and are being addressed. Dr. Winn is a nationally recognized, internationally recognized authority in access to therapy. You've got to have good therapy to have access to, but in addition, there are other issues that we must figure out how to overcome, not only in stem cell and cellular therapy, but in even more routine therapy. Dr. Winn, could you comment on this issue of access and how we get this fantastic technology into more and more patients with cancer?
First of all, thank you all for having me as part of this panel. I always enjoy being at this conference. I learn a lot, and it's just a fantastic conference. I want to apologize for my absence there. I would like to just let you know that Donald McEachin, who is a U.S. congressman, unfortunately succumbed to GI cancer, colorectal cancer, and I was at his funeral. And so I apologize for not being there in person, but it got me even more motivated about access issues. The interesting thing is when I think about access issues, I think about technology. And in fact, I've been going back through the literature and looking and with a fine-tooth comb and figuring out that, interestingly enough, with almost every new technology or every new screening intervention or medical intervention, we inadvertently increase disparities. It's a wonderful concept about what I think our challenges of the 21st century is bringing both the discovery phases and the implementation and access phases together in a way so that as we have these wonderful new therapies, we don't do what we've done in the past. An example of that is breast cancer. For many of you who are historians, you can recognize and see a trend, particularly with the Ciurea data of the introduction of the mammographies and the inadvertent introduction of disparities between Black and white women. The most interesting part of this for me and why this discussion is meaningful is because as we even look in the context of who are on clinical trials, particularly with CAR T therapy, we see a disparity. In fact, back in 2016, 2017, there was a number of articles written about the immunotherapy divide, which was real. The nice part about this story is that, like any other big science question, the issue is if that's the struggle, then how do we fix it? Or how do we deal with it? I think we're finally starting to understand that there is some aspects to certainly the bench to bedside model that has served us well. But one of the aspects of that is actually finishing that from the bedside into the community. And I think that what we've learned getting through this COVID period and others is that in addition to what we do at the bench, what we do in the context of community education, getting beyond this concept of community outreach to real community engagement matters. And in fact, I recently, the AAMC just posted their 10 principles of trustworthiness, academic or institutional trustworthiness. And one of the first ones is that the community does actually have education and does have knowledge. In some cases, that's why they don't trust us. I think that we are going to have to work a little bit about understanding that it's not so much that many of these communities don't want to participate. It's just that we have not followed the principles of, one of our brilliant folk of thought leaders of W.B. Yeats, who said, think like the wisest of men and women, but communicate in the language of the people. Frequently, we have gotten ahead of ourselves, which is fantastic to have the amazing science. But then the question becomes, for example, if minority populations are doing less well, how then do we then communicate in the language of the people effectively? What I've been recognizing as a cancer center director is that I am as enthusiastic of what I'm doing at the bench and the science that evolves at the bench. I'm equally enthusiastic of the science that's now coming out of how do we better communicate, how do we better engage, and I dare say, how do we better integrate our community so that we're bringing the science closer to them so that they will actually have access. I think I'll end with sort of saying that some of the access issues, certainly, we always sort of say to folk that minorities don't want to be on clinical trials. It turns out that there's now, that's old literature. New literature is suggesting that that's actually not entirely true. when, communicated effectively, many of minority populations and communities want to be part of a trial. But you do actually have to get over some elements of being able to effectively communicate. There is a level of education that's not only part and parcel with minority communities, but also rural communities as well, which we don't talk much about. Having said all that, I am enthusiastic and optimistic that after these last several years, we are getting much more of a sense that we actually have to do great at the bench, but we also have to do even better in the context of communicating effectively in our communities. And so many of my efforts here at Massey has been able to bridge the discovery and the implementation sciences and the community in a way that Harold P. Freeman and others did this way back in the 90s. This is really, I think we're on the age the age of a 2.0 version of that ability to get people to these cutting edge, high impact, and meaningful therapies.
Thanks very much, Dr. Nguyen. And you remind me of a conversation I had over dinner last night with Ms. Patricia Wiley, one of the founders of the patient advocacy movement in this country, that in order to have individuals participate on trials, you first have to be partnered with individuals in the community. I think this was a concept slow in evolving in many areas in not only oncology but other areas of medicine. But I'm pleased to say that I think there is real progress being made, but we got a ways to go. In that regard of ways to go, while we don't want to focus inappropriately on cost, Dr. Grupp has thought a lot about how you implement these wonderful therapies, but also particularly the economic barriers. Could you talk a little bit about that, Dr. Grupp?
Yeah, and there's always this natural tension between having the appropriate resources, especially to our pharma colleagues, to develop new medicines, and what that costs the healthcare system. And I'm no healthcare economist, but I am certainly aware of the fact that we spend somewhere around 17 to 19% of our national income on healthcare. So that's a lot of money. And so the access issue, I mean, from a doc's point of view, we talk about diversity, equity, and inclusion, but that translates to diversity, equity, and access for us. And so I'll take a couple pieces of that. One is, going from clinical trials to an FDA-approved product does improve the potential for access. But in the cell therapy world, what we do is complicated, has a lot of regulatory overlay. You're, collecting cells from a patient, sending them off, getting them back. It's kind of a big deal. And right now, that's really only available in large medical centers. So that's an access barrier. And people are trying to make this a little bit easier and a little more accessible, but it's really not going out to the community quite yet, and it may be a little while before that happens. Problem one. So just as an example, at CHOP, 85% of our patients come from outside our catchment area, across the whole country and to before the pandemic, the world. And what we do is we have sources of money to at least get the patient there with their caregiver, put them up for the time that they need to be in Philadelphia. That's something, and it really helps a pretty good fraction of people for whom they couldn't come without that support. But if you got a job where you can't be away for the afternoon, you can't be in Philadelphia for 6 weeks. So that's an area of access that I worry about. And that's something we're trying to address. But then, yes, let me touch on the price issue. I mean, we can't get around the fact that these are extraordinary prices. So in 2017, when Ciurea was FDA approved, a clinical trial, which I was the head of the study steering committee for Novartis for, that was approved at $475,000, which at that time was the most expensive single cost therapeutics available in the US formulary. And I'm like, great. And you can argue that price. It's very complicated and expensive to make these cells. We want to make them cheaper. All those things are true. But there it was, $500,000. And the reality is that looks quaint now. So the Bluebird product for thalassemia, which is a blood disorder that's related in a general way to sickle cell disease, and we're going to see the sickle cell products come out next year. That's FDA approved. $2.8 million, and then we've just heard about an infused gene therapy. Doesn't involve a cell, but some of the similar technology for hemophilia, a bleeding disorder, doesn't involve a cell, $3.5 million. That's the top right now, and I don't know where that goes from there. And the issue is if you have commercial insurance and these are relatively rare disorders, you get them approved. But what happens when this stuff does start working in solid tumors, as Stefan mentioned, and we're talking about much larger incidence populations, and sickle cell, again, non-cancer, 100,000 or more patients with sickle cell in the United States. What do we do about access under those circumstances? And then this gets back to Dr. Nguyen's comment, and I'll stop with this, about the community. So for the last 10 years, I've been really focused on how do I get people to a large medical center and support them in that journey and get them back to their doctors where they're actually treated every day. And I think that's an important thing to think about. But now in Philly, we got a lot of sickle cell patients who could potentially benefit from this. And how do we reach out to the community to actually get them engaged, have the information they need to understand what's going on, and then get them access just to the folks in Philadelphia who might potentially benefit from this. And so that aspect of cost, especially when you get away from commercial insurance and more toward Medicaid, is, I think, a really concerning barrier. And that's where, I'm not going to argue what it costs to be on Gleevec, the drug for CML, for 20 years. I'm not going to argue what it costs to be on some of the newer targeted therapies for a year. It comes out to about the same kind of money. They're all very expensive, but fundamentally, especially when it's a one-time hit, when you're not talking about $2 million over 10 years, but you're talking about $2 million on Thursday, then that's a challenge that we are going to reach in larger incidence populations and in patients who aren't as well insured. And look to us over the next year or two and see what we can do in working with the Bluebirds and the Vertexes of the world to try to address these issues, and hold us accountable if we're not doing the right thing.
Stefan, did you want to say something?
Yeah, I have two comments actually, and I must say that practicing medicine, I realize how the barriers are at the insurance level. So I find often that there are different levels. I'm not familiar with very well with the system, but it turns out that if you have a better insurance, or if you're a better insured for a particular insurance, you have access, for example, to clinical trials. So if you do not have access to a clinical trial of this nature, let's say. That's one consideration. The second consideration, I think it's truly ironic that these therapies that are now the most expensive therapies on the planet are geared towards most disadvantaged and most socially disadvantaged and economically disadvantaged individuals. So I think this is going to be, this is probably this is overall the most challenging situation, because third-party payers are are on one side try to limit their their expenses, and how we are going to have these newer drugs accessible to this population that is where it is needed, in fact. So it's very difficult to picture, for me at least right now, how this is going to pan out with these extraordinarily high costs. And one last thing, if I want to comment on, is because I reflected after we had a brief chat before on the actual cost, and apparently the cost of a therapy is not in any way run by any government agency. So there is, it seems to me that these costs are set by the companies alone. So there is absolutely, seems to, I don't know, maybe I'm wrong, there's absolutely
You're not.
There's absolutely no oversight. So there is not a panel, a government agency that says, let's review how you came up with these costs for these therapies. So,
well, you touch on an important concept particularly important perhaps for in the Nixon Library, the library of a very skilled and internationally known politician. You're touching on some very politically sensitive areas that we're going to have to solve. But Dr. Grupp mentioned the drug Gleevec. We think of the terrific expense of cellular therapies and bone marrow transplantation. We forget that economics played into the early development of drugs like Gleevec, small molecules as well. The development of Gleevec for chronic myelogenous leukemia was slowed by Dr. Brian Drucker's challenges, Dr. Drucker, the discoverer of Gleevec, Brian's challenges in getting drug companies interested in developing a small molecule drug that would be expensive to develop and only available for 4 or 5 or 6,000 patients a year, a very small market. And many drug companies passed on that opportunity. Drug companies obviously have come around, but Linda, have you thought about this issue and small molecules?
They're coming around slowly. They still are waiting for the next platinum, or something that you can give everybody, and it's not going that way. Especially going with the precision medicine pieces now, where you might very well be some tiny subset of somebody with leukemia or a tiny subset of somebody with breast cancer. The whole field is going this way. It's no more dumping just one big vat of one drug. You're going to have therapy cocktails of 2 and 3 and 4 different drugs at a time. So they're going to have to start thinking of more of a bar content. The bar is now serving this particular cocktail. They're going to have to think their way through that. It's not just the big bucket of one kind of drug. They're going to have the manufacturing is going to have to change as a whole. I feel for them, but it is there is a paradigm shifting, but it's slow.
I have to say, can I say that that's why I'm excited
in developing your small molecule?
Well, I didn't think about that. I was chasing a target, making a drug. But even the prices of clinical trials are so very expensive. I started my own company in order to finance the clinical trials that are needed to move the drug forward. Clearly, there are going to be a ton of pressures for the cost of therapeutics. I mean, this is it's where we're going.
Absolutely. One thing we absolutely don't want is limit innovation. So this is absolute, and you're the perfect example here. We really don't want to limit innovation, but on the other side, it seems like there is a huge gap to how what it was mentioned before, how we actually end up giving to many people that need the new therapeutics. So this is a huge challenge, I think.
But an exciting one. But an exciting
Dr. Winn, do you have
Absolutely. This is why I'm super excited. I'm excited about the science. I'm excited about not only bringing in the populations. As you're having these discussions, I was thinking about some of the recent work that got done in Montefiore and other places that showed when, in this case, when African Americans and Latinos got access to the CAR T therapy, their outcomes were statistically, there's no difference between black-white outcomes, et cetera, et cetera. But the cool part about what we were just talking about is the thoughtfulness behind the precision medicine movement. The thoughtfulness is that, for example, for me, that a woman, and I'm really glad you said what you said, is that the days of just giving one cisplatin and everybody, that those days are probably moving further and further away. And we're moving into really this new, really innovative, cutting edge space in which we're personalizing the medicine we give. That's important. I'm going to say because, for example, the work that's been done by Melissa Davis and others, say in triple negative breast cancer, has shown us that if you're Black, that's what we call it, right, which we know is a social construct, but more of Eastern African descent, you have a much more favorable outcome from triple negative breast cancer than you would if you were Black with Western African ancestry. I actually think that we've moved. The funny part about the field is not only is the basic science moving to, I think, a higher level and being much more nuanced and much more rigid and much more focused and refined, but this issue about who we're treating and who's going to be impacted by the precision medicine we give is also evolving in the context of not only understanding one's ancestry, that's the first level, but also understanding the impact that one has of place and space on their genome, right? I've always said that how the genome, that is the ZNA, that means the zip code neighborhood of association, also plays a role into your responsiveness to therapy. It's going to be ridiculously messy, but I'm excited about the mess. Because just like immunotherapy over 120 years ago, I think you just had to have bright people continually to think about how to solve the problems. And then you get that aha moment, as you sort of said, the 20-year delay, where someone else comes along and says, aha, now it works. I actually think the bridge between the DNA and the ZNA, this sort of interplay that we are, particularly in the context of precision-based medicine, is going to matter. And by the way, I think that and I'll shut up, but I do think that there's one other thing that we ought to think about that we may not be able to deal with here is the criteria that includes or excludes people we ought to probably revisit. Because I don't think we've talked about those standards, about those inclusive and exclusive criteria and how they also inadvertently exclude many people who are minority without really good validation for why. So I think we're in this new frontier, if you will, exciting about where precision medicine, person-based medicine will fit into how we're going to do this for not just some, but for all. And that's the complexity of it, but that's the excitement of it as well.
And it is particularly exciting to me to hear Dr. Nguyen commenting on his enthusiasm for dealing with a mess, because we certainly have a mess. Linda?
Actually, another point, well, we talked about costs and community. Another concern, a part of the mess, if we want to go there, is actually the education piece. I mean, we have to educate the entire medical culture now. I mean, that's, we have you have this incredible knowledge, but, most everybody's out there in the community, and now we have to educate all the medical community for these type of therapies.
That's a terrific segue into perhaps an interlude where we can open the floor up to questions from the audience to ask for clarification on any of the points that have been raised. Are there questions anyone like to ask any of our experts? Yes, sir.
Hi there. I'm Flavio Roque. I'm actually representing Dr. Drucker from OHS Unite Cancer, so I'm glad this conversation came up. And I think it's very good points that the panel presented. I mean, you're talking about treating a rare disease and then the economics of that for a pharmaceutical company. But the interesting about the Gleevec story, of which, as you know, we celebrated the 20th anniversary of the drug, is that not only did it work for CML, but then there was this other disease, gastrointestinal stromal tumors, where the same mutation played a role, and similar situation, about 6,000 patients a year, not a huge number, but revolutionized the treatment of that disease. And so I think that's the other interesting piece, that you may find mutation one disease site and have it applied elsewhere. So fantastic discussion. Thank you very much.
Other questions or comments? Identify yourself, if you would, please.
I have a question that I would like to pose to you, which you just brought up so eloquently. I believe that perhaps we should question the federal government's expenditure of welfare funds to non-citizens, and shouldn't that money be redirected back to our own citizens? In other words, let's examine where our funds are actually going. And this seems like such a much more worthwhile direction for those of us that have relatives and friends that are minorities.
So I'd like to quickly give a slight perspective on that. And I have respect where that question's coming from, and I think the issue of resource allocation is incredibly important. But, you know, I'll give you a different point of view, which is I'm a pediatrician. I'm a pediatric oncologist. And if I have a patient in front of me who has cancer and I have the ability to save their lives, that's what I'd like to do.
Thank you. Terrific issue, but complicated, and I respect very much Dr. Grupp's perspective. Other questions?
Hi, my name is Patricia Wiley, and I'm a patient advocate. And I just wanted to say, I want to echo what Dr. Winn said, that things are changing now. And there are so many other groups that we need to look at. And one of the most important things I think you need to look at is the cultural aspect of those groups and what goes on, not just the color of their skin, but what their culture is, because of a lot of it would really surprise you. So I encourage all of you who work in oncology to please have your community advocates, your patient advocates, delve into that, because that's critical.
Yeah, as an old guy who's been around for long enough to see the transition, it is remarkable to look at the degree or the lack thereof of community engagement in the 80s and early 90s with cancer centers and their communities, and how much that has changed in the last 10 to 15 years, to no small extent from the hard work of folks like Patricia, who have really made us realize how critically important it is. Other question?
We're going to take one last question.
Yeah, my name is Luhua Mahmoudou. I'm representing Cal State Fullerton. Yeah, so this is actually a very interesting discussion, and I think one factor in which we have to take into consideration is the time to the treatment of the cancer after the individual have been diagnosed with the cancer. Yeah, because we actually had a research which we were actually looking at the time for which an individual started treatment of the cancer and what are the factors that actually influences the time the individual start their treatment for the cancer. Being able to know these factors which influences the time the individual start the treatment of the cancer after they have been diagnosed is very important in actually mitigating some of the factors when it comes to the survival of the cancer. So I think it's something we have to take much consideration. Thank you very much.
Good point. Question or comments from any of the panelists on that important point?
It was just a quick thing. And first of all, I just want to tell Patricia that I am a huge fan. So I wish I were there, because this would have been outstanding just to see you in person. But short of that, I will say that when it comes to issues like time and frequency and cadence, particularly of the trials of getting the therapies, this is where I think the bigger issues are going to come in a context of understanding how the impact of the social determinants of health play a role. It also is going to actually be incumbent upon, I would say, that the big pharma companies and the academic universities and other health systems in figuring out how to streamline the process a little bit to actually help us not only get people on the trials, but to do it in a timely manner. So I think it's again, another complicated aspect that I have had immense joy, believe it or not, over the last couple of years trying to figure out how to get people, to treatment, and then how to actually do it where we're reducing the time to activation. I will say that probably not in this setting, but there are things that we're now working with other groups around the country to figure out how we can do this better. But transportation and other issues like that seem to be of no consequence when we're talking with people with resources, sometimes are the small things that get in the way of getting many of our people, particularly that we need, on the trials, getting the therapies, and getting it in a timely manner. So I will tell you that thank you for that question, and that there are at least a handful of us around the country really putting a lot of thoughts into how to come up with a system to make it better.
It's real quick. The other aspect that I am so struck by is, so I work at Children's Hospital Philadelphia. It's a great place. But it's a typical quaternary care hospital, and getting through the system, although we try so hard to make this better, getting through, it's not just getting to the hospital, although transportation is a huge barrier, and at least that seems like something you can address. But once you're there and you walk in the door, the process of getting through the system, not only, dealing with your insurer, dealing with the hospital, dealing with the 42 places that you need to go, dealing with the registration process, do you have the app? I mean, all of those things are incredible barriers to care for people who don't have the prior experience to allow them to deal with these circumstances. I was treated for lymphoma 10 years ago. You want an honest answer? My wife took care of most of this stuff. And when I saw what she went through to get my care, it was incredible, and we're somewhat plugged into the system. So that aspect of just dealing with the complex systems that we've built, which are really not built around the patient, they're built around other considerations. Dr. Winn, tell us how to fix this.
We probably don't have enough time, although I bet Robert could summarize it in a small length of time. But we should probably thank the organizers of the conference, a wonderful opportunity for us to get together. And particularly, I want to thank my co-panelists for a stimulating discussion and excellent questions. So thank you all for your attention, and it was a great pleasure for us.
Thank you.
Thank you.